Hello everyone. Here, MSc Medical Biotech students perform experiment of recombinant DNA technology at Dr. D.Y. Patil International University under the guidance of Dr. Priyatosh Ranjan Sir. Following is the list of experiment performed. Now, let's start with our experiments.
Isolation of plasmid from EcoRI GM101 using alkaline lysis method. Firstly, sanitizing hands using a 70% ethanol solution. Then labeling all solutions and tubes. After that, the culture that has grown overnight is being poured into a microcentrifuge tube. Then centrifuging at 10,000 by 12,000 rpm for 5 minutes. Removing the supernatant from the tube. Adding 150 ml resuscitation buffer, solution 1. The solution is being vortexed thoroughly. Right now, we are adding 200 ml of denaturation solution, solution 2. Following that, 150 ml of neutralization solution, solution 3, is being added. Store the tube on ice for 3 to 5 minutes. After that, centrifuging at 10,000 by 12,000 rpm for 10 minutes. The supernatant is being taken and transferred to a new 1.5 ml microfuge tube. Currently, we are adding an equal volume of phenol, chloroform. As of now, the organic and aqueous phases are being mixed through vortexing, and subsequently, the emulsion is being centrifuged at maximum speed for 5 minutes in a microfuge. After the centrifugation process, a 3-layered formation is currently observable, and the upper layer is being transferred to a fresh nucleic acids are currently being precipitated from the supernatant by adding to volumes of ethanol at room temperature. Mixing the solution by vortexing and then allow the mixture to stand for 2 minutes at room temperature. Collecting the precipitated nucleic acids by centrifugation at maximum speed for 5 minutes at 4 degrees Celsius in a microfuge. Completely removing the supernatant. Adding 1 ml of 70% ethanol to the pellet. Recovering the DNA by centrifugation at maximum speed for 2 minutes at 4 degrees Celsius in a microfuge. Removing all of the supernatant by gentle aspiration. Adding 50 ml of nuclease-free water. Conforming the purity of plasmid DNA using agarose gel electrophoresis. We are preparing a 1% gel, which means we need to measure 1 gram of agarose and add it to 100 milliliters of 1-extras acetate EDTA buffer. Heating the solution for dissolving it completely. After cooling down the solution, we add 6 ml ethidium bromide having concentration 0.5 μg per ml and then slowly pour the solution into the tray. After the gel solidify, we transfer the gel to the electrophoresis chamber and add tris acetate EDTA buffer till the gel is deep into it. Now loading the 24 ml sample to the wells. The power supply was switched on till the bromophenol blue reached the anode and then removed the gel slab and visualized under UV light. Purification of plasmid DNA was successfully done.
Restriction digestion of PUC19 using EcoRI restriction enzyme and analysis using agarose gel electrophoresis. Some restriction enzymes leave the DNA with uneven ends, which is also known as sticky ends. The discovery of the restriction enzyme that could cut the DNA at the specific site made the genetic engineering possible. The restriction enzyme found naturally in bacteria EcoRI. This restriction enzyme binds the recognition sequence GAATTC. It cuts the serval phosphodiester bond of the DNA and makes sticky ends. For this activity, we need samples of nucleus-free water, 10x assay buffer, 100x BSA, PUC19 and EcoRI. Then we label and thaw the solution before use. Firstly, we are transferring 41.5 ml of nucleus-free water into the new tube. Following that, introduce 5 ml of 10x assay buffer into the mix, which helps to maintain the pH and enzyme stability to ensure the efficiency of the DNA cleavage. Then, adding 0.5 ml of 100x BSA, it acts as a stabilizer and can improve the efficiency of the restriction enzyme digestion of impaired DNA. Now, adding PUC19. This PUC19 DNA is the target DNA which we want to digest. Finally, adding 1 ml of EcoRI. It is a restriction enzyme which recognizes the specific DNA sequence, that is GAATTC, and leaves the DNA at those sites. Now incubating the vial at 37 degrees Celsius for 2 hours. Restriction digestion of plasmid was done successfully with EcoR1 and a single linear plasmid band is observed in well 2 to 7. In well 1, one uncut plasmid was loaded and its band observed above the linear plasmid. Hence, uncut plasmid is present in nick or open circular form of conformation. Restriction digestion of PUC19 using BamHI and Ssp1 restriction enzyme and analysis using agarose gel electrophoresis. Inside the bacterial cell, there is a circular piece of DNA known as plasmid. Here we are using PUC19 and there is a special enzyme called as the restriction enzyme which recognize, find and cut the specific DNA sequence which are used to open up the plasmid. Now here we are using two different enzymes, BamHI and Ssp1. BamHI creates the sticky ends whereas Ssp1 creates the blunt end. Here they form two fragments. One is of 604 base pair and the second one is 2082 base pair. For this practical, we need nucleus free water, 10x assay buffer, 100x BSA, PUC19, BamHI restriction enzyme, and Ssp1 restriction enzyme. First, we are transferring 11.5 ml of nucleus free water into a new tube. Following that, introducing 2 ml of 10x assay buffer into a mix which helps to maintain the pH and the enzyme stability to ensure the efficiency of the DNA cleavage. Here added 0.2 ml of 100x BSA. It acts as a stabilizer, increase the efficiency of the restriction enzyme digestion of impure DNA. Now adding 4 microliters of 20 Ng per ml of PUC19. PUC19 DNA is the target DNA which we want to digest. Now for the digestion we are adding BamHI restriction enzyme which creates sticky ends. Finally adding the second restriction enzyme which is SSP1 which creates the blunt end. Then we have to spin down the vial for a few seconds. Then the vial was then incubated for 1 hour 15 minutes at 37 degrees Celsius. To confirm the digestion of plasmid, we have to perform agarose gel electrophoresis. This is the result in which we load our sample of group 1 in well 4. Here we observe different conformation of plasmid. Two intense bands at 604 base pair and 2082 base pair are visible which indicate that double digestion had taken place successfully. To purify and quantify the gel extracted plasmid DNA by UV visible spectrophotometer. For the purification and quantification of the plasmid DNA, sodium salts are used to enable the DNA binding to the silica bead. Washing step removes the impurities and elution buffer with the sodium salt release the purified DNA. DNA concentration is determined by its absorption of light at 260 nanometers. We begin by weighing a small vial which is initially weighted 1.1 gram. Then we carefully cut out two 6-8 base pair DNA band using a sterile plate and after that we reweight the vial. Now its weight is 1.7 gram, which means the weight of gel is 0.6 gram. Next we added 1.5 ml of sodium iodide having concentration 6 molar to the tube. The tube was then placed in 55 degrees Celsius incubator for 2 to 3 minutes to dissolve the aqueous gel. 15 ml of silica beads were added and kept at room temperature for 10 minutes. Then the tube was centrifuged at 10,000 rpm for 30 seconds. Then we carefully collect the supernatant from the tube without disturbing the pellet. To further clean the DNA, we have to add 200 ml of wash buffer to the tube. Wash buffer contains 3 molar of sodium acetate in 75% of ethanol. Then centrifuge at 12,000 rpm for 30 seconds. Once again, we collect the supernatant in a tube. Then again we add 200 ml of wash buffer to the tube. The tube was then incubated at 55 degrees Celsius. Now 25 ml of 1x TAE buffer is added to the pellet. The mixture was kept at 55 degrees Celsius in dry water bath. Following that, the mixture was centrifuged at 10,000 rpm for 1 minute. and the resulting supernatant was collected in the fresh tube. Now, the gel was extracted successfully. To check the purification, the purified DNA was quantified with the UV spectrophotometer. For this, we dilute 10 ml of purified DNA with 2990 ml of distilled water. Absorbance was recorded at three wavelengths: 230 nanometer, 260 and 280 nanometer. Quantification of the gel is done successfully with the help of UV spectrophotometer.
To perform blue-white screening in bacterial cell. The molecular mechanism of blue-white screening relies on lag-Z reporter gene. In this technique, the non-recombinant colonies with the function of lag-Z gene encoding for the beta-galactosidase turn blue in the presence of cultural media containing. Let's start the procedure. First of all, we have to take 100 ml of LP broth, add 100 microliters of 100 mg per ml ampicillin in both, then 100 per ml of IPTG and 20 mg per ml of Xgal. Stir it well and pour the media into the Petri plate. Now firstly we have to take overnight cultural media. Then the culture was equally distributed in two tubes. The tubes were centrifuged at 4000 rpm for 10 minutes at 4 degrees Celsius. The supernatant was discarded. Now the pellet was resuspended by adding 5 ml of ice cold zero point 0.1 molar of CaCl2 in each tube. Again, centrifuge at 4000 rpm at 4 degrees Celsius for 10 minutes. Now, the supernatant was discarded without disturbing the pellet. Then we have to resuspend the pellet with zero point 0.1 molar of CaCl2 and glycerol adding 800 ml in each the tube. Arrange the vial on the ice. 100 microliters of culture was collected in two 1.5 ml of vial. Now 5 microliters of ligated sample and 5 microliters of control added to separate 100 microliters of the component cells. Both the tube were incubated on ice for 30 minutes. Then both the tube were placed at 42 degrees Celsius for 90 seconds. And then the tube were immediately kept on ice for 10 minutes. 800 microliters of LB media was added to both the tubes. The tubes were kept at 37 degrees Celsius for 45 minutes in a shaking incubator. Centrifuge for few seconds. Now the 100 microliters of transform and the control cells were spread plated on Xgal IPTG Lurica agar plate containing ampicillin. Both the plates were incubated at 37 degrees Celsius overnight. Blue-white screening was successfully performed. We observed blue and white colonies both on the culture plate. Molecular cloning of green fluorescent protein in Escherichia coli. Green fluorescent protein cloning involves the insertion of the GFP gene into a target organism, which is used as a visible marker. For making LV broth, we have to add 1.5 gram of agar powder in 100 ml of distilled water. Add 12.5 gram of LB in 500 ml of distilled water. Add 400 ml of LB in 6 gram of agar. Now add 100 ml of LB to 1.5 gram of agar. Autoclave it. Now pour the media into the Petri plate and allow it to solidify. For the ligation reaction, the components were added in a sequence as in the table. First we add 11 ml of nucleus-free water, then 2 ml of factor having concentration 3 nanogram, then 4 ml of insert DNA having concentration 7 nanogram. 2 ml of 1x ligation buffer and finally 1 ml of T4 DNA ligase, and then we have to vortex it. The vial was kept at 4 degrees Celsius overnight and the broth was kept on the shaker. On next day, the culture were transferred equally into tubes. Then centrifuge at 6000 rpm for 8 minutes at 4 degrees. Then discard the supernatant completely. Now 1 ml of ice cold calcium chloride solution is added. After resuspension, 8 ml of calcium chloride is added. Then the tubes are kept on ice for 20 minutes. Centrifuge for 20 minutes at 3500 rpm. Then discard the supernatant. Again, we have to add 850 ml of calcium chloride. Adequate 100 ml of suspension in 8 tubes. For the heat inactivation, the ligated sample were kept at 65 degrees Celsius for 10 minutes. Now add 100 ml of competent cell in the sample and incubate on ice for 20 minutes. For the heat shock treatment, both the tubes were kept at 42 degrees Celsius on water bath for 2 minutes and on ice for 6 minutes. Then add 500 ml of LB broth to both the vial and incubate at 37 degrees Celsius for 1 hour. 100 ml of each vial transform with the ligated mix onto the LB plate and spread it well. Incubate at 37 degrees Celsius for overnight. After exposure to UV range, the colonies didn't glow and the ligated product is not formed. It could be because of the insert was not ligated in the vector or the orientation of the insert was wrong. All these experiments were done in DYPIU labs under the guidance of Dr. Priyatosh Ranjan Sir. Thank you.